Alright, so everyone, today we are going to learn a very important concept in hematology and oncology physiology, that is the antiglobulin test, which is also to be known as the Coombs test. Right, so we can begin up this session by understanding one of the major questions, that are the antibodies reacting against our red blood cells or not? Right, you have to remember it out that antibodies attach to the red blood cells, and those red blood cells may be destroyed, leading to hemolysis, the anemia, the jaundice, and sometimes a dangerous transfusion reaction, right? So that's why it all does matter. But by the end of this lesson, you should clearly understand two things, the two important questions that I have to understand majorly in this one. The first understanding must have to be circulating around that how the direct Coombs test helps diagnose the AIHA. What is that? The autoimmune hemolytic anemia, right? And the second thing you have to understand, that how does the indirect Coombs test helps prevent the transfusion reactions, right? So let's just begin off with one of the fundamental ideas, that the red blood cells normally circulate freely in the blood, but sometimes antibodies bind to the red blood cells. When the antibodies bind to these red blood cells, they can mark them for the destruction by the immune system. So the Coombs test helps us detect that these anti-red blood cells antibodies. The Coombs test helps us detect the presence of antibodies which are circulating against the red blood cells over here. Okay, this is the major concept or fundamental one that you have to remember it out. Now you have to remember it out that we do have the two different types. The first one, we do have the direct antiglobulin test, and another one is the indirect antiglobulin test, or you can also say the direct Coombs test or the indirect Coombs test. Okay, the direct means that antibodies are directly attached to the red blood cells, and indirect means that antibodies are in the serum. Okay, now let's look up to the figure over here at the top. The figure tells us that the antiglobulin test detects the antibodies against the circulating red blood cells. Okay, this one. And so for this one, you have to remember it out that the test that we do use over here, we do use the Coombs reagent. Okay, the Coombs reagent, and this is also to be known as the anti-human globulin, right? So this part is important because the Coombs reagent is not the patient's anti-red blood cell antibody. Instead, it is an antibody against the human antibodies. And what's the main job? Its main job is actually to bind to the human antibodies that are already attached to the red blood cells and make the clumping visible. This clumping is known as agglutination. This clumping is known as agglutination. Let me show you over here. This one, agglutination, right? So you have to remember it out that agglutination means antibodies are present. Okay, agglutination means the antibodies are present. Now, if you just look up to the left side of the figure, this is the direct test. Okay, over here, this is the left side. So the direct test that you are looking up over here, which is also known as the direct Coombs test. Okay, so the question which is above the left side, it says that does this patient have autoimmune hemolytic anemia or not, right? So we have to break down it all once, right? The autoimmune means that the immune system is attacking the body's own cells. Hemolytic means the red blood cells are being destroyed. Anemia means there's reduced red blood cell mass or the reduced hemoglobin. So in AIHA, the patient's immune system produces the antibodies against the patient's own red blood cells. And these antibodies attach to the surface of the red blood cells inside the body. Then the immune system recognizes those coated red blood cells and destroys them, often in the spleen. Okay. So clinically, this patient may have fatigue, the pallor, the jaundice, the dark urine, okay, and increased radiculocytes, and some of the signs of the hemolysis too. Now, over here, you have to remember it out that when we take the patient's red blood cells, let's say that we have taken the patient's red blood cells, okay, and then we add the anti-human globulin or the Coombs reagent to this one. This means that we are checking whether the antibodies are already stuck on the patient's red blood cells or not. So if the antibodies are present on the red blood cells, the Coombs reagent binds to them and links the red cells together. Okay, and all this linking of the red blood cells, while after adding up the Coombs reagent, the test will be considered positive when there is agglutination being present over here. Okay, it means that there is already the presence of the anti-red blood cells antibodies. Okay, so this is to be known as the positive Coombs test, right? And this supports the autoimmune hemolytic anemia. And if we just look up to the negative results under the direct test, there is no agglutination. You see that? No clumping of the antibodies with the red blood cells. It means that the antibodies were not detected on the patient's red blood cells, so the test is negative and also known to be as the negative Coombs test. Okay. Now, here is one of the curious clinical questions that you need to remember it out. That why do we need the Coombs reagent at all, right? If the antibodies are already on red blood cells, why can't we just see them clumping? So the answer is that many clinically important antibodies, specifically the IG, the IgG, right, the immunoglobulin, the G1. So many of these antiglobulins, they are small. They can coat the red blood cells but may not bridge them strongly enough to cause visible clumping by themselves. So Coombs reagent act like a bridge and it binds to antibodies on the nearby red blood cells and bring the cells together, making the agglutination visible. So you can just think of the Coombs reagent as somewhat like a revealer, right? It reveals the antibody-coated red blood cells. And now if you just move towards the indirect antiglobulin test, which is the indirect Coombs, right? Over here, you can just start up by asking one of the questions that will the recipient react against the donor red blood cells or not? That is the major another concept that we are going to cover it up. So this is mainly about actually blood transfusion safety, the blood transfusion safety. Okay, so before the blood transfusion, all right, what is we need to know that whether the recipient semer contains the antibodies that could attack the donor red blood cells or not. If the recipient has the antibodies against the donor red blood cell antigens, giving that blood could cause a transfusion reaction. So in the indirect test, we are not asking whether the antibodies are already attached to the patient's red blood cells. Instead, we are asking, are there free anti-red blood cell antibodies in the patient serum or not, right? So now over here, you can just see that we have taken the patient serum, right, with or without the anti-red blood cell antibodies, okay? It means that after all this one, we have added anti-human globulin and also the donor blood. Okay, so this part specifically actually interprets that if the patient serum contains antibodies against the donor's red blood cells, those antibodies will bind to the donor cells. Then the Coombs reagent definitely going to actually cause the agglutination. And so definitely the visible clumping will be seen over here. And what is this actually going to say up? That the patient or the recipient serum has the antibodies that reacted with the donor red blood cells. In practical terms, that donor blood is not actually suitable because the recipient may attack it, right? So now look at the negative result. It shows the no reaction and there is no clumping at all, no agglutination. This means what? The recipient serum did not react with the donor red blood cell. And that is definitely a very good sign for the compatibility, right? So you have to remember it out, the both of them ones, okay? Used for the pre-transfusion testing, right? And over here, if you just look up, let me over it up. Yeah, over here you see that. Okay, the first thing, anti-human globulin is added. Yeah, anti-human globulin, which is also known as Coombs reagent, added to the patient's red blood cells. The red blood cells agglutinate if the red blood cells are coated with the anti-red blood cells antibodies and used for autoimmune hemolytic anemia diagnosis. The indirect one, normal red blood cells added to the patient serum. If the serum has anti-red blood cell antibodies, red blood cells agglutinate when the Coombs reagent is added, and this is actually used for the pre-transfusion testing, right? You have to remember it out. One of the question, which is the clinical one, may usually ask that why is the indirect Coombs test important before the blood transfusion? So the answer to that one is that because the recipient may have the antibodies from the previous transfusion, the pregnancy, or the exposure to the foreign red blood cell antigens. These antibodies may not cause the symptoms while they are just floating in the serum. So if the incompatible donor blood is transfused, they can bind to the donor red blood cell and cause hemolysis, right? So the indirect Coombs test helps prevent a dangerous transfusion reaction before it happens. So that is why it is being actually done. Okay, I hope so that both of them are clearly understandable, right? The direct and the indirect one. There are some of the questions that we need to actually understand that. Let's say that if you do have one of the patient which has anemia, jaundice, and the positive direct Coombs test, what does that suggest? You see that the result is the positive Coombs test, and we do have the patient with the anemia, the jaundice. Okay, so what does that suggest? I give you a while to think of that one. You can pause the video to think. And for the ones, if you're already get to the right ones, you have to remember that it suggests that the patient red blood cells are coated with the antibodies, and these are causing the immune-mediated hemolysis. And one important diagnosis is definitely AIHA for this one. Now, let's say another question that a patient's serum reacts with the donor red blood cells before transfusion, and which test is positive? So definitely that is going to be the Coombs test, that is going to be the positive one. Another one, they may ask you that in pregnancy, which test can screen the mother's serum for antibodies against the fetal red blood cells? That is also, again, the indirect Coombs test, right? To check it out. And if the newborn's red blood cells are already coated with the maternal antibodies, which test would detect that? That will be the direct Coombs test, because antibodies are already attached to the newborn's red blood cells. So that is why many students over here, they actually confuse the direct and the indirect, because both tests use the Coombs reagent and both can show the agglutination. But so do not memorize based on only the reagent. Both use Coombs reagent. Instead, memorize based on the sample. In the direct Coombs, the patient red blood cell is being used, and in the indirect Coombs, that the patient's serum is actually being used out. I hope so that this is absolutely clear to you guys.